Hello, everyone. My name is Bhavya Vats. I'm a second year biomedical engineering major. My research mentor is Dr. Harata in the Department of Molecular Physiology and Biophysics. Today, I'll be presenting on effective image segmentation for analyzing dendritic branching of neurons. Our goal is to be able to compare protein expression of single neurons. Both the conventional neurite tracing method and our hybrid masking hope to isolate the cell of interest from a large field of cells. We will accomplish this by eliminating unwanted signals from the background or neighboring cells in the image field to isolate the signals from our cell of interest. Once the cell is isolated, we can perform further analysis to compare protein expression levels of disease state versus wild type cells. As one example, our focus for this presentation will be on mouse striatal neurons, and we will be looking at fluorescent microscopy images stained with MAP2. That's the microtubule associated protein, which looks red in our images and highlights the neuronal dendrites. We will also compare one of the conventional methods of image segmentation of neurons, known as the Simple Neurite Tracer, with our proposed method of hybrid masking. We define masking as the process of removing signals that don't belong to our cell of interest. The Simple Neurite Tracer is an open source ImageJ plugin used to segment neurons by manually tracing or drawing the dendritic processes. Here we show the original image with background noise and signals from neighboring cells. This second image shows the traced dendrites in pink, and the final panel shows the output image of the filled neuron. There are two key disadvantages of tracing neurites for segmentation in this fashion. First, the output image includes the positive signals from the cell of interest and also from the background. This can be seen by plotting the pixel intensities versus distance in microns across these yellow lines um, for the original and the post SND images. On these line plot profiles here, this sharp peak corresponds to this dendrite here. The maximum intensity or the Y value of the peaks for both graphs is the same, indicating that the final output image contains signals from the cell of interest and also from the background. Another downside of using SND is that it's hard to detect the edges or the end of the dendrites when tracing the dendritic processes by hand. This is especially difficult for weak and irregular processes. To overcome these drawbacks, we propose the hybrid masking method, which combines ImageJ tools with our MATLAB program to mask or to remove unwanted positive signals. We apply the ImageJ Gaussian blur filter to the raw image to subtract a blurred or background image with a large standard deviation value from our raw image with a small standard deviation value. That is why it's called the difference of Gaussians. The JB result image does not include background noise, and the dendritic structures have clearer edges. We can see this graphically by plotting the pixel intensities versus distance across these yellow lines for both the original and the JB result images. The Y values for these peaks for the JB result image is smaller as compared to the original image, indicating that we have successfully eliminated the background. The next step is to manually remove the unwanted processes really close to our neuron. For example, we remove the structures in blue um, to be able to draw the dashed yellow line within our MATLAB program. After drawing this region, our program excludes the positive signals that are outside of the marked region, giving us the post pre-processing image, which is also oriented by the apical dendrite or the thickest and longest dendrite to the 12 o'clock direction. To summarize, the simple neurite tracer method is quick to use and is user friendly. However, it includes the background noise and fails to include weak and irregular signals. It is also harder to detect the ends of the dendrites when using SND. Comparatively, our hybrid masking approach successfully excludes background signals and signals from neighboring cells, and we can also manually choose to include weak and irregular dendrites. It's also easier to detect the edges in hybrid masking after performing the difference of Gaussians. That brings us to the end. Thank you.